Okay, yeah, perfect. Thanks for the introduction. So yeah, exactly. I'm going to be talking about my PhD project, which has focused on looking at sex differences in human genetic architecture. So a little bit of an introduction. You may know that males and females are different, but what I think a lot of people don't realize is that these differences are quite expansive. They expand just for reproductive function or that way that we look. So, for example, males and females have been reported to respond differently to different kinds of medication, as well as to present different prevalences in certain illnesses like mental illness or cardiovascular disease. So, what underlies these sex differences is truly not very well understood. People think sex differences and they think the sex chromosomes, and they would be correct in that these do play a very important part in sexual differentiation. But once a human has been completely differentiated at birth, um, truly there are so many different things that could be contributing to the differences that we're observing. So beyond your genetics, and more specifically here genetic architecture, which is just a fancy term of referring to how your genetics leads to your phenotype, and that can be anywhere from your alleles, your epistasis relationships, dominance relationships, gene by environment. All of these things could be contributing. But in addition, there have also been differences reported in epigenetics, differences reported in gene expression, as well as in gene expression regulation, differences in hormones that could be contributing, another classic factor that has been well studied. And then another thing that people don't really think about is environmental exposure differences that could be arising because of societal gender roles that could also be influencing some of these things. So, for example, if females are more predisposed to go into a particular line of work and this line of work has a particular risk, this could potentially also influence the risk of a particular disease. However, if we really wanted to simplify it, you could say that there's your genetics and then there's everything else in this classic gene plus environment equals phenotype framework. Except here, our environment would be everything that is defined or correlated with our sex. So things like epigenetic differences, things like genome regulation differences, and things like hormones, all of these things could potentially be interacting with our genetics to lead to some of the variation in phenotype and some of the differences in phenotype that we're observing. So essentially, this is just a framework to think about the problem. And it's very similar or it's identical to a gene by environment interaction study, whereas here the environment is sex. So it's a gene by sex interaction that we are studying. So how do we go about studying gene by sex interactions? Here I've arbitrarily summarized the different ways that we can do that into the broad view and the narrow view methods. Now with the broad view, what we are trying to do is we're trying to establish the relationship of the genome as a whole to the phenotype. So for example, by measuring how heritable a trait is in a measurement that is very aptly named heritability, we are able to estimate how much of the phenotypic variance can be accounted for by genetic variance. So if we were to calculate heritability in males and in females separately, and we were then to see a difference in heritability between the sexes, this could indicate that there is a certain heterogeneity in the genetic control of that trait between the sexes. Another way to study gene by sex from a broad perspective would be to obtain genetic correlations, like Joanna was just talking about. So a genetic correlation is, in broad terms, a way of describing how common the level of genetic control is between two groups, two diseases. So here, the genetic correlation that we would be interested in would be between males and females for the same trait. So a genetic correlation that is close to 1 would, like a normal correlation, indicate a level of high common genetic control, whilst a genetic correlation of 0 would indicate no common genetic control. So a genetic correlation that's significantly different from 1 would then indicate that there is a certain level of heterogeneity in the genetic control of a given phenotype between males and females. Moving on to the narrow view methods, I think in computational biology, this is probably the most popular way of studying, or what is most commonly known, and that is genome-wide association studies. Now, in contrast to the broad view methods, genome-wide association studies allow us to dissect the genome bit by bit and ask how each individual genetic variant is associated to the phenotype. So if we were to then perform sex-stratified genome-wide association studies, we would then be able to estimate how each individual genetic variant is associated to the phenotype in males and females separately, and then ask, are these associations significantly different based on our sex? For it may be that a given variant might be contributing to a phenotype in just one of the sexes, or it might be contributing differently depending on our sex. So if you look at the overall literature pre me starting my PhD, you would find that differences did exist, but they weren't reported to be very big. Authors had also described the need for larger statistical power and to study a wider breadth of traits, because while it is true that some traits like height had been well studied in genome-wide association studies, others had not really focused or narrowed down genome by sex interactions. And also, as well, we need to understand the phenomenon, because many times in a genome-wide association study, you might find an association, but you don't really know what it means biologically. So our aims with this study were, first of all, to search for gene by sex interactions across the genome, establish the extent across both the genome and the phenome, and then also to shed light on potential mechanisms on how these gene by sex interactions could explain some of the differences observed in phenotype. We tried to assess the effect of gene by sex on the transcriptome, as well as to explore underlying mechanisms. So to do this, our main data source was UK Biobank, which consists of around 500,000 white European individuals. After quality control, we ended up with 530 traits, which were both binary and non-binary. Binary traits consist of disease traits mostly, so whether or not you have had a particular disease, whether or not you have had diabetes, so on and so forth. And so we also after quality control, we ended up with around 9 million genetic variants in our analysis. So this is a pretty big amount of data that we were then able to exploit to our advantage to analyze gene by sex across both the genome and the phenome. So even though the methodology here was quite varied, truly at the heart of everything, we had sex-stratified models, and more specifically, linear models, and more specifically, even more, linear mixed models, which then allow us to quantify relationships between our genetics and our phenotype. And by running sex-stratified models, we were able to do this for males and for females separately in a way that then allowed us to compare and assess whether they were significantly different. So essentially, what we're trying to do with these models is just quantify the relationship of both your individual genetic variants to the phenotype, as well as the relationship of the genome as a whole to the phenotype, using linear mixed models, as I've already mentioned. So on one hand, linear mixed models are incredibly valuable to us because they allow us to control for population structure. They also allow us to obtain variance components of phenotypic variance, so we can obtain estimates of the genetic variant component of a phenotypic variance, as well as environmental variance, and this then allows us to obtain heritability estimates. Then on the other hand, it allows us to estimate the genetic effect of each individual genetic variant here with a beta I've described, or a BF or a BM. It allows us to describe each genetic effect to each of the 530 phenotypes that we are considering here. So once we've established and fit our models, we began to ask the questions whether we could find gene by sex interactions, and how, and what does this mean? So the first question that we wanted to answer was, is there a difference in heritability? So again, a roundup is that heritability is the fraction of variation of a trait that can be explained by the genotype. In rough terms, it's a little bit more complicated. But essentially, we found that 30 of the 446 binary traits did have a significant difference in heritability between the sexes, whilst this was around 50% for the non-binary traits, which is a pretty large amount of traits. We then moved on to genetic correlations that significantly differed from one, which again is the level of common genetic control between two groups. Here, we are considering males and females. Now here we were a little bit more limited because we were using LD score regression, which uses GWAS summary statistics to obtain genetic correlations. And it does recommend that you use traits with at least 5,000 cases, which did limit our analysis for binary traits to just 77. But even still, we found that 11 of those 77 had a genetic correlation that significantly differed from one. Whilst for non-binary, it was 58 of the 83 that had a genetic correlation that significantly differed from one. Now here I've included a histogram which includes the value of genetic correlations, and the height is the number of traits that presented said genetic correlations. And this is just the traits that presented a genetic correlation that did significantly differ from one. So you can see, even if they did differ from one, the majority are concentrated close to one, meaning the level of common genetic control is quite high between the sexes. This parallels what had been reported already. However, we do see some traits that do differ more. So you see things like disorders of the urinary tract, hernia, eczema, gastric reflux, waist-to ratio representing the non-binary traits, and so on. So this is still quite interesting. Now moving on to potentially the most interesting part was our sex-stratified genome-wide association study that allowed us to dissect each individual bit of the genome and ask which areas could potentially be interacting more so with our sex and factors correlated with our sex. So To do this, what we did is we compared each individual variant and we assessed which genetic effects were significantly different between the sexes. And if we did find a significant difference, then we would call that variant a sexually different variant or an SD-SNP. And so I'm going to refer to those as SD-SNPs from now on. And essentially, we found the traits that had at least one sexually different variant were 61 of the 84 non-binary traits and 42 of the 446 binary traits. Now here I've included for both of the categories those traits that presented the largest amount of hits or independent SD-SNPs after accounting for LD structure in the genome. You can see that by far waist-to ratio is the trait that had the largest amount of independent SD-SNPs, but we also see other things like height, trunk fat percentage. And then for the binary traits, we see traits like ankylosing spondylitis, gout, hypothyroidism, which is quite interesting because we know some of these diseases do present more in one sex than the other, gout being more prevalent in males and hypothyroidism being more prevalent in females. So now that I've talked about that, we've seen that gene by sex exists. Now, what are the actual consequences of this? How can this affect our day-to-day GWAS analysis? Well, on one hand, you might not know, but genome-wide association studies are what underlie prediction or polygenic scores. So genome-wide association study effects or models are used to then intake new individuals. You take their genotype data, you just input them in your model, and you are able to obtain a prediction of the phenotype that you are studying. However, it could be in the situation that we're observing here, if there are significant differences between the sexes, it could be that by fitting sex-specific models, we might obtain a better prediction for males and for females. This could be because, for example, you might have genetic effects that are negative in one sex, positive in the other, and thus by combining the two, you are not getting an optimal genetic effect estimate for one sex or the other, and instead they could potentially even cancel each other out. And linking to this, another consequence that we wanted to test was whether in genome-wide association studies that include both males and females, we're ultimately missing information of interest for just one of the sexes. Because it could be, for example, that a sex-specific factor might be binding to a particular SNP, but it's sex-specific, meaning that this particular SNP or a change in allele in this particular SNP might only be interesting or associated to a given phenotype in just one of the sexes. So this is what we are calling masking. Starting off with what we wanted to test was whether we could get an improvement in prediction. What we did is we retrained our models, but matching sample sizes. Because even though we already had fit our models, if we wanted to compare models that compared the effect of a sex-stratified model versus a non-stratified, we wanted to match sample sizes so it would be a more fair comparison. So to do this, we reran our genome-wide association study models with 150,000 females for the female-specific model, 150,000 males for the male-specific model. And for the agnostic model, we took 75,000 males and 75,000 females, and we combined those, and that was our sex-agnostic model. Now, once we had our genetic effects for those three distinct models, we wanted to test whether we could improve prediction. So to do that, we obtained phenotype predictions for each individual in our testing data set in three different ways. First, we obtained the same polygenic score, or the same prediction, by which if you were female, then to obtain your prediction, we would be using the genetic effects from the female-specific model. If you're a male, then we would use the genetic effects from the male-specific model. The opposite would be the opposite, like the name indicates, where if you're a female, then we would use the genetic effects from the male model. If you're a male, those for the female model. And then finally, the agnostic polygenic score, which would represent what is currently done, in that independently of your sex, we would just be using a genetic effect that has been estimated in a GWAS that includes both males and females. So to assess whether we could get an improved prediction, or we assessed the prediction accuracy by obtaining a correlation of the estimate or the prediction with the real phenotype value. So we only tested traits that had at least 10 SD-SNPs because these polygenic scores were only estimated using SD-SNPs, not all GWAS hits. So this is why we have correlations here represented on the y-axis that are very low, because you can't really get a very accurate phenotype prediction based on a very low number of SNPs, especially if you have very polygenic traits. So what we saw was that there was no significant difference in the prediction ability. However, we did see an increased trend of prediction accuracy. So here you can see our opposite predictor did by far the worst with both males and females, followed by the agnostic, which lies somewhere in the middle. And the same does seem to do better across all of the traits that we looked at, which are the non-binary traits with at least 10 SD SNPs. Now, this could suggest that as larger sample sizes become available, and as we characterize gene by sex interactions further, we could potentially get better models to predict when we are considering sex stratified models, but this is yet to be seen. So moving on to our test of the masking and on non-stratified analysis, again, I'll remind you that by masking, we mean that it could be that a variant is significant in one of the sexes, but not the other, and thus by combining the two together in a sex-agnostic model, you might end up with a non-significant signal. So to test this, we did exactly that. We looked for variants that were significant in a female-specific model or that were significant in a male-specific model, but that ultimately led to a non-significant signal in a sex-agnostic model. And we found evidence of at least one potentially masked SNP across a wide range of traits. So for the binary traits, this was 98 of the 446 traits. For the non-binary, it was even larger than that. It was 78 of the 84. Now, what this could be suggesting is that we really should be performing sex-aware analyses where even though, yes, by combining both males and females together, we have larger sample sizes. And it does make sense to do this because generally speaking, genetic architecture is shared. And so by increasing sample size, we are increasing the power to discover variants that are interesting. But it also could mean that it is worth looking at sex stratified models, or at least including an interaction term in their models to make sure that we're not missing anything that could be sex specific. Okay, so I've talked about the existence of gene by sex, and I've talked a little bit about the more immediate consequences. But what does this mean? Like I mentioned before, genome-wide association studies are kind of a black box. So we don't really know many times what this means. Why are these gene by sex interactions arising? We don't really know. So to shed light on this, we did two things. The first thing was a gene set enrichment analysis, by which we looked for patterns in the genes that fell nearby our SD SNPs. So like a gene set enrichment analysis indicates, the first thing that we did is we translated our SNP data to gene data. So we mapped our SD SNPs to nearby genes. And with those genes, we then performed gene set enrichment analysis. Furthermore, we took our genes and we separated them based on the genetic effect of the SNPs that mapped to these genes. If the SNPs that were mapping to these genes on average presented a larger effect in females or a larger effect in males, we divided those into two, those that had a larger effect in females, those that had a larger effect in males. And then we did the set enrichment with those two groups of genes for each trait. Now, what we wanted to do with this is see if we could get information on sex-specific pathways that could inform some of the difference that we had been observing. And we used FUMA for this. And sadly, we didn't see any commonality. We saw huge amounts of heterogeneity and pathway variety while looking at the traits that we ran through this gene set enrichment analysis. So truly, there was no one-size-fits-all explanation for gene by sex. It wasn't that all of these genes had a binding site for a particular hormone or a particular sex-specific factor, or at least thought that we had found. So it is interesting. And it is quite interesting that there is no obvious link to steroid hormone regulation, which I guess is what we were expecting. However, if you look at the literature, you will find that people have described that sex differences in gene by sex do arise irrespective sometimes of hormone regulation. And the GTEx cohort, the paper that came out in Science where they looked at differences in gene expression between the sexes, they also describe sex biases in gene expression and gene expression regulation. And they find that these genes, only a third of them actually had binding sites for hormone response elements. So it is quite interesting. And it does point to this being a little bit more complicated than we had previously thought. So speaking of the GTEx cohort, we used the GTEx cohort to see also if we could link our SD SNPs to gene expression. So to do that, we took our SNPs and we looked at nearby genes and we asked whether a change in allele of these SNPs could be leading to a difference in gene expression regulation. This is what is called the sex bias EQTL. And so here I've represented a little diagram explaining what this would entail. So again, for example, this could occur if a particular SNP falls within a binding site for a sex-specific factor, then a change in allele would truly only affect gene expression in one of the sexes, but not the other. So we used the GTEx cohort for this, which consists of 39 tissue types and gene expression and genotype data. And it's incredibly valuable because many times we only have access to blood gene expression data, but because we have SD SNPs arising from a variety of different phenotypes, we don't really know what would be the appropriate tissue to be looking at. So this gives us a very holistic view. However, the downside of GTEx is the sample size. So we did find 8 sex-biased EQTLs, but truly larger sample sizes are needed to unravel this connection. And also because we expect these effects to be fairly small in the same way that GWAS effects are fairly small when you have very polygenic traits. So the take-home messages here are that, yes, gene by sex exists. It's small, but it's quite widespread. There is potential improvement in trait prediction, but that is yet to be seen as larger sample sizes become available. Potentially more interesting is the case of masking, which makes a case for sex-aware frameworks and making sure that we're not missing things by completely ignoring the fact that sex is there. It was quite interesting to see that we didn't find an obvious link to hormonal regulation. So again, that's yet to be seen and discovered further, as well as a link to gene expression regulation, because the sample sizes sadly were not there. So I wanted to leave the presentation with a bit of hope in that there is still so much left to do in this area to help us understand why there are so many differences between the sexes in phenotypes. So for example, sex awareness will bring huge amounts of insights into these studies. We also haven't really looked at into rare variants because we have been using data from the first releases in UK Biobank that are based on array data. So we're not really looking at whole genome sequencing or whole exome sequencing. That will also bring light to rare variants and what they're doing. Sex chromosome methods can be improved. Also, here we're only looking at UK Biobank, which is heavily white European, meaning that we can't really say how gene by sex occurs across other populations. We're also looking at a very narrow point in our life because UK Biobank are usually older individuals. This means that we are truly only focusing on gene by sex on the older population. So we're only really looking at postmenopausal women as well as older males, and we know that hormones are likely playing at gene by sex, are likely leading to some of these interactions. So it wouldn't be crazy to think that gene by sex is also a function of time, and it will be really interesting to see how it varies from time period to time period in everybody's lives. And beyond that, single cell insights, multiomics, and better causal inference and fine mapping will also give clues into everything that we're saying. But with that, I'd like to acknowledge the work that went into this. I'd like to thank my supervisors, both Alberta and James, and then a huge amount of people that helped me throughout the years at the Roslin at the University of Edinburgh. And if you want to know more, here is the publication. We also made available all the GWAS summary statistics. So if you have any questions, I'd be happy to answer.
Great, thank you, Elena. Another wonderful talk. We already have a question from RJ Murray. Would you expect a copy number variation to impact the mapping? And if so, do you have an idea of how it would impact it?
So from what I've read, there is a small number. I believe that when I was reading the literature, they said that there was a couple of copy number variations that did differ between the sexes. But in this particular study, we're only looking at SNPs, I'm pretty sure, or like variants that have two alleles. So we didn't really go into copy number variations. There are a couple, though, that have been described to differ between the sexes, but they weren't very, very many from what I recall.
Okay, from Joanna, thank you for a very clear presentation. I was wondering about the heritability differences. Do you generally see higher heritability in one sex versus the other?
Yeah, so that's a great question. No, we looked for patterns such as if there was always a dominant sex over the other in terms of heritability. And no, we didn't really see that. It is quite interesting because in the past, there was this hypothesis that stated that there was this, I believe it's called the sexual protective hypothesis or something. It essentially means that it could be that you need a higher amount of liability in one sex versus the other because you have more protection for whatever reason, and that would essentially entail a higher heritability in one sex versus the other. But that is not what we saw here. It doesn't seem to be the case, no.
I have a question, I guess. In our experience, sometimes the phenotypes in the UK Biobank don't necessarily follow the SEER statistics of what we might expect. And I think, there's probably some bias in it. Mm-hmm. Of course, every human database is going to have some of these limitations. I guess, what other data sets have you thought about potentially applying this to? And I guess, what do you think needs to happen? What is the biggest barrier?
Yeah. So I think one of the biggest barriers is what I said is at the moment, for me, at the minute, the main factors that come to mind are, population. So at the moment, we're only looking at white Europeans, and this is the case across a huge amount of genetic studies, and it's because it's what we have available, but it's truly not a big representation of the human species. So that is on one hand, and the other is what I also mentioned about age. So we know that there's diseases that change prevalence at different sexual reproductive times because hormones play a huge role in our biology. So, for example, I believe it was asthma that it's more prevalent in prepubescent females and then it shifts, and then also cardiovascular disease. It seems like females are more protected until after menopause, and then that shifts again. So it's things like these that, mapping gene by sex across the human lifetime would also offer very interesting clues, I think.
Yeah. I mean, given its limitations, UK Biobank does have a lot of measurements and phenotypes cataloged for individuals, where I guess our typical GWAS studies are very focused on a particular phenotype and don't give you that rich kind of
Yeah, there's a huge amount of, I guess, interesting cohorts. You have, I guess, FinGen is a great resource at the moment. 23andMe are collaborating with a bunch of people, and they have a huge amount of data as well. I'm currently working with Generation Scotland, which is here in Scotland. So I hope I get to play around with that and also look at sex differences in that. So yeah, it's really exciting. And also beyond, Europe and the US, there's a lot of other cohorts that could shed light into the whole population differences thing as well.
Thank you. It's very exciting work. Emilio, do you have a question?
Yeah, I have a curiosity. So at some point you mentioned that when you do the analysis, you just focus on the SD SNPs. So this means that you remove many SNPs. I'm wondering if, for example, you can build something that is somehow intermediate, where you actually, depending on the phenotype you're looking for, you can select a pool of SNPs that still can be important, because maybe you can recover some epistatic interaction between the sex SNPs and the other SNPs that are important to explain the phenotype.
Yeah, I guess there's a thing that happens with genome-wide association studies where you have a hard threshold and you say, oh yeah, this SNP is significant or this SNP is associated to this phenotype. It doesn't necessarily mean that the rest are not associated, but we are under this pressure to say, do we have enough statistical evidence to say that this particular loci is truly associated? So it is very, what we wanted to do here is kind of give a general overview of everything. But truly now it falls on, I guess, the experts in each individual disease to kind of dig deeper and find what SNPs are exactly truly associated by fine mapping, assess causality, all of these things. And what, like you said, it's truly not black and white, but I guess that's the thing with genome-wide association studies. Yeah, I guess that's a bigger problem, I think, than we realize, whether a SNP is significant or non-significant. But yeah.